Hello, and welcome back to Human Physiology. So now that we've had a pretty good understanding of the ligand-receptor relationship, let's talk a little bit about an application of that. Agonists and antagonists and their role in pharmaceutical drugs. So we've said before that the relationship between the ligand and receptor is not unlike the relationship between a key and a lock. So the general idea is that a ligand needs to have exactly the right shape in order to fit into the binding pocket on a receptor. But this is not strictly true, even though that is a very good analogy to kind of go ahead and commit to your memory. So what is reality is that ligands that are structurally very similar to what we'll call the natural or normal ligand, these types of ligands are still capable of binding to the receptor and somewhat eliciting a response. It may not be the full response. In some cases, it may even be well beyond the sort of response we might expect. But these types of molecules that are not the actual primary or natural ligand but still activate the receptor, these are called agonists. So you can see in this diagram here, on the far left, we have the natural or primary ligand that binds to the receptor and gives us the natural physiological response. An agonist, it's not going to be exactly like the natural ligand, but it is very much going to structurally and shape-wise resemble that ligand. So it should still be able to bind into that binding pocket and give us a range of different responses. Maybe not quite as good of a response, or maybe even better. So that is an agonist. The other type of molecule is going to do just the opposite. If you have a ligand that can bind to the receptor but not give you any sort of response at all, then you've got an antagonist. But even that description doesn't really do it justice. There's something that we're missing here. So an antagonist will bind to the receptor, as you can see in the picture here, but it's not just that it will not give us a response. What it's also going to do is it is going to block the receptor, meaning that any natural or primary ligand that is in the vicinity will not be able to bind or engage that receptor as long as the antagonist is bound. So if you kind of picture in your head here that you've got this receptor that is blocked by this antagonist drug, then even if you have a whole bunch of natural ligand out here in the extracellular fluid, that's not going to do you any good because you don't have any available receptors. That receptor is blocked by the antagonist. So the reason why we want to at least be aware of both agonists and antagonists is because when you move on to work in a clinical or hospital setting, pharmaceutical drugs are going to be kind of the lifeblood of patients on the road to recovery, right? So what pharmaceutical drugs are agonists and antagonists. So the idea here is that over the course of decades, research scientists have studied these pathways, studied the way cells respond to natural ligands that our body produces, so hormones and neurotransmitters and things of that nature. They have studied these receptor proteins so that they know exactly how those ligands bind. And what these scientists have done is they have designed drugs that very much resemble the natural ligand, but will either act as an agonist to turn on or keep on a certain pathway, or they design an antagonist which will block a certain pathway that we do not want on. So the idea here is that in a lot of human diseases, say for one instance, a disease might be caused by a receptor that is keeping a certain signal transduction pathway on when that pathway is not supposed to be on. So in that case, an antagonist drug could be very useful. We can use an antagonist drug to block that receptor and keep it from keeping that disease-causing pathway on. Alternatively, if we have a receptor that is off when it is supposed to be on, we can use an agonist to get it to bind to the receptor and turn it on. So whether a certain pharmaceutical drug should be acting as an agonist or an antagonist, it's just going to depend on the situation. There's no hard and fast rule that says agonists are good for this and antagonists are good for that. So a perfect example or application of this are beta blocker drugs, which I'm sure that you have heard of before. So beta blockers are antagonist drugs. These are drugs that block the beta receptors that we talked about before, the beta receptors that epinephrine binds to, except these beta receptors are located mainly on the heart. So the idea of how a beta blocker works is that typically during a fight or flight response, epinephrine will raise your heart rate by binding to those beta receptors on the heart. Well, what that's going to do is that's going to drive up your blood pressure. And for some people, some vulnerable people, whether it's the elderly or someone with a pre-existing cardiovascular condition, we may not want that. We may not want them to get excited and their heart rate and blood pressure to go up. So if someone takes a beta blocker drug, we can prevent epinephrine from doing this, raising the heart rate and raising the blood pressure, by blocking those beta receptors on the heart and preventing epinephrine from having any sort of effect. So by doing this, we can keep the heart from working itself too hard, and we can keep the blood pressure nice and low. Yeah, and these last two sentences here kind of sum that up. So one last thing that we'll go ahead and discuss here, this doesn't necessarily have to do with agonists and antagonists, although you certainly could apply this here. Sensitivity to a ligand. So as you can probably imagine, even though we haven't come straight out and said it just yet, how sensitive a cell is going to be to a particular ligand is going to depend on how many receptors there are available to bind to the ligand. If a cell only has one receptor, it doesn't matter if there's billions of ligands in the extracellular fluid, only one ligand is going to be able to bind at a time. So it's going to be how many receptors are capable of binding to a ligand that's going to determine how strong of a response we're going to get. So cells actually have a mechanism for sensitizing themselves or desensitizing themselves to ligand based on how much ligand is available. So the first method of this is called receptor upregulation. This is how a cell is going to become more sensitive to ligand in cases in which there may not be a whole lot of ligand around. So by synthesizing new receptors, so this is going to kind of bring us back to a lot of the chapter 3 stuff on protein synthesis. There is a gene in the DNA, instructions on how to build this particular type of receptor. That gene has to be transcribed into mRNA. The ribosome has to get that mRNA and translate the protein. The protein's got to fold into its shape and be translated in the rough ER. We've got to package it up in the Golgi, and then we've got to export it to the cell membrane so that it can be stuck in the membrane. So all of that stuff still applies here and kind of summarizes why the stuff that we covered in chapter 3 is still important. Okay, so this is the mechanism here. If the cell wants to become more sensitive to a particular hormone or some other type of ligand, the cell can decide to turn on that gene, synthesize more receptor, so that over time, as you go from left to right here, we have more and more and more receptors that are available to bind to a particular ligand. So the more ligand we can get binding to more receptor, the stronger and stronger and stronger a response could come. The alternative is what is called receptor downregulation. So this is going to be reserved for cases in which there's way too much ligand. Basically, there's so much ligand, we're keeping these receptors on and activated all the time. And usually the cell is not going to like that. In these cases, the cell is going to try to desensitize itself in an effort to get this pathway to turn off. It's basically been on for too long. If you've grown up in a household with kind of a stingy father, and I think all of us have been there before. Oftentimes, kind of your prototypical father is the one that kind of goes around the house turning off all the lights saying, oh, electricity isn't free, you know, right? So we've all kind of been there. So that's kind of what this is. Basically, there is so much ligand around, the cell basically says, okay, the lights have been on for way too long. These receptors have been activated for way too long. Let's save some energy. Let's shut this process down. So what the cell is going to do here through downregulation is internalize some of these extra receptors by endocytosis and then deliver it to the lysosome to be destroyed. So over time, as these receptors are destroyed and internalized, fewer and fewer and fewer receptors are now available to bind to ligand. So since there are fewer receptors, the response strength gets smaller and smaller and smaller. So a good application of this in kind of the real world is adaptation to painkillers. So if you know anyone that has been prescribed painkillers like Vicodin or Percocet for a very long time, the longer that they're on these drugs, no matter how valid their prescription is or how valid their need is for it, the longer they're on these drugs, they start to feel like the old dosage that I've been on for forever, it's just not cutting it anymore. I don't feel the benefit that I used to. So the idea here is that if they're taking painkiller every day, that painkiller is a ligand. It's one of those pharmaceutical drugs that we talked about. So that ligand is around all the time. It's bound to receptors all the time, and cells just naturally don't like this. So they try to take the receptors away, meaning that the strength of the painkiller response gets diminished more and more over time. So a person's natural inclination is, well, the dosage isn't working anymore. I need to take more painkiller to feel the same effect. You're basically forcing extra ligand to occupy what few receptors are available. And then we're not going to get into the specifics of any of these diseases that are listed here, but this is just kind of an on your own if you're interested in receptor-linked diseases, meaning diseases that are caused by some faulty ligand-receptor relationship. There are some diseases listed here. So diabetes insipidus is a defect in the vasopressin, or what's more commonly called antidiuretic hormone receptor in the kidneys. So it causes issues with water retention and urination. Familial hypercalcemia, so this is going to be calcium sensing proteins in the parathyroid. Retinitis pigmentosa, whooping cough. So these things are all caused in some way by some sort of defect in a signal transduction pathway that is precipitated by that ligand binding to a receptor. So I'm not going to ask specific questions about any one of these particular diseases, but these are all very good places to start if you're looking, say, to learn more about a particular disease, or alternatively, if you just feel like you need some practice in examining or interrogating the ligand receptor relationship and you want to see it actually in action here. So for any of these, if there is some defect in the ligand receptor relationship, the idea here, depending on the application or the context here, whatever it is, if there's a defect, then what should have been the natural physiological response, it's either going to be diminished or it's not going to happen at all. And as we've seen, anything that happens in physiology is all about maintaining homeostasis. So if something's not going right here, then homeostasis is not going to go right, correct? All right, so that is going to do it for this particular video. So we only should have 2 more videos to cover in this particular video series on communication. Neither of them should be too terribly long. So starting next time, we are going to revisit the homeostasis response loop, and we're going to learn a little bit more about sensors. That's kind of the one thing that we have not really known a whole lot about. So we are going to explore that next time. I invite you to join us, and I will go ahead and see you next time. Bye-bye.